Now that we're all here, welcome and good evening. My name is Sherelle, and I will be your host for this evening's educational session. I work as the marketing manager for Reha Care, and with me tonight is my colleague Anne Hughes, who will address any sales or support inquiries you may have after this webinar. We appreciate you taking time out of your evening to join us. We will hold the Q&A at the end of the presentation, but please feel free to type in your questions in the Q&A inbox at any point in time during the presentation, and we will address this at the end of Brett's presentation. And for those of you interested in learning more about low-level laser therapy, Brett has made time in his very busy schedule to deliver 5 hands-on based seminars this year. The seminars will include demonstrations on cutting-edge functional neurology, neuroorthopedic rehab, and low-level laser applications. So please keep an eye out for the invitations to come or visit our website and go to workshops and events for more information. Without further ado, please welcome Brett Jarosz.
Thanks, Sherelle, for the introduction. Evening, everyone. Thanks for joining us on a Tuesday evening. I will just get my screen up here for everyone. So focus of tonight is looking at low-level laser therapy and the relationships with mitochondrial function. And when we get into the literature, really the primary thing here, if we look at what low-level laser therapy does, it's got everything to do with the mitochondria. So for those that didn't attend or review any of the YouTube webinars that are available from Reha Care from last year, we did a series of 3 webinars where we just built the scientific background, some of the introduction and history behind low-level laser therapy, and then we moved into more of some of those clinical applications and then into some more advanced clinical applications, also differentiating the different types of photobiomodulation. So that low-level laser therapy is one of those modalities within photobiomodulation. And we tried to use those webinars to help us separate and understand the differences between LEDs, low-level laser therapy, high-intensity laser therapy, infrared, etc. So we'll hit a few of those points today, a little bit of expansion, but for further detail, feel free to go and review those webinars that'll be made available from Reha Care for those in attendance. So disclaimers, as always, I'd like to just state for everyone, I'm not an employee of Erconia or Reha Care or any other product or vendor that I talk about in any of my presentations. I just am a consumer of their products that I utilize within my own clinical practice. The presentation tonight is not about diagnosing, treating, or curing any medical disease process. Everything I'm always trying to do is about supporting the normal adaptive physiology of the human body. Everything that I try and present, whenever I do present, I'll always try and have every slide referenced where I can, so that way you know where the information's coming from and you can go back and fact check that information. Any of the treatment protocols, if we ever talk about them, not tonight, won't be about treatment protocols per se, but anything that's off-label in any of my presentations, understand that even though that off-label aspect may not have a specific FDA clearance or approval for it, knowing that there's always evidence to support the thing that I'm trying to present. Depending on practitioners that are here tonight, always remain within your scope of practice. Obviously, understand what your rules are for your state, jurisdiction, for your profession, understanding whether or not laser therapy, photobiomodulation, high intensity LED, whatever the terminology is, please make sure that if you're going to be using this type of technology within your practice, that within your particular profession, within your state, that falls under the guidelines for APRA for you in that region. So what I want to try and cover tonight is looking at wavelengths. So when we say wavelengths, we're talking about the color of light. Let's just keep it that simple. So the color of light and the relationship with the mitochondria, how those two interact. Then we want to look at breaking down that mitochondria to the electron transport chain, obviously how that produces ATP, understanding how issues with the electron transport chain or issues with the mitochondria, if we have dysfunction there, how that can influence human health. And then we want to start to look at how different wavelengths influence the components of the electron transport chain. And then we want to look at the different things within the literature that have been shown to have therapeutic effects as it relates to photobiomodulation and specifically low-level laser therapy into mitochondrial function. So part one, looking at, again, color of light and the mitochondria. So if we look at the literature, research about the mechanism of low-level laser therapy inevitably involves the mitochondria. So everything keeps pointing back to that as its function, and it's attributed to the absorption. Keyword, absorption. We have to absorb the light in order for it to be able to have a physiological effect. So the key thing, though, is the absorption of monochromatic visible light, so like one color. And what we're going to see as we go through the presentation is the different parts of the electron transport chain that we can see in the picture there, complexes 1, 2, 3, 4, 5, or 5 ATP synthase, is if we look at those different complexes, we know that those complexes, each of those seems to absorb a certain color of light. And that absorption, well, I shouldn't say it seems to absorb, if the light can be absorbed, then that light that's absorbed, different color light, influence different complexes within the mitochondria. So that appears to be the mechanism of how all light therapy works. So those effects, when we can get that absorption into those different complexes within the mitochondria, we start to see increased proton electrochemical gradient, more ATP synthesis. So we can see there RNA, protein synthesis, oxygen consumption, membrane potential, enhanced synthesis of NADH and ATP. But let's really keep it simple. I need to absorb the light. We know certain colors are absorbed in the complexes within the mitochondria better, or the colors have a better effect on certain complexes. So the color of light influencing the complex 1, 2, 3, 4, and the end result is better functioning mitochondria, better production of ATP. All right, so take-home message, absorption of a single color of light and better production of ATP. So the first law of light therapy, which you've already heard me say a few times here, is the first law of photobiology states that for low power visible light to have any effect on a living biological system, the photons must be absorbed. So we have to absorb the light, and then these mechanisms become important. So on the picture there on the right, we can see the schematic of the idea of a red colored light. So the wavelength there in that sort of 600 nanometer range is where that red color is being absorbed into the mitochondria. And the one that you can see pictured there is cytochrome C oxidase. So you'll see that referred to as COX or CCO in abbreviations. So that's part of complex 4. So red light, hint already, red light is absorbed in complex 4 because of cytochrome C oxidase. So cytochrome C oxidase is basically a molecular photoacceptor. All right, so it's accepting that color light. And the key bit in point 2 there, photons that are delivered into living tissue can either be absorbed or scattered. So for it to start to have its biological, physiological effect, it has to absorb. If a photon scatters, so idea if it doesn't get absorbed into the tissue, if it comes across and hits something and scatters off, it can either deflect out via diffuse reflection, or later on that deflected or scattered photon can then be absorbed by another tissue over there. All right, so scattered photons will eventually be absorbed or will escape from the tissue in the form of diffuse reflection. So a couple of important points here, and again, just setting this scene, the difference between visible light, so when we think of the rainbow and the colors that we can see, versus infrared light. And especially now, this idea of like infrared saunas and infrared heat lamps, and emphasize that point, heat lamps. We understand the mechanisms here. So when we talk about low-level laser therapy, So one of the Erconia devices here that I have, an older version of the EVRL, the low-level laser therapy works via photochemistry. So the visible light that we can see on me here, that visible light has a photochemical effect. So visible and infrared affect biostimulation in different ways. The visible light elicits photochemical changes, whilst infrared can only produce physical changes. So understanding that, although visible light can produce photochemistry, infrared radiation only produces molecular rotations and vibrations. So think of it like a microwave. We put food into the microwave, we turn it on, and those microwaves, those invisible light waves as such, they cause this molecular rotation and vibration. Infrared does this to our water molecules. So infrared goes in to the human body if it's absorbed. And when it gets absorbed, it's absorbed by water. So infrared is absorbed by water molecules, and it makes water molecules oscillate and vibrate, and it creates a photothermal effect. Hence, now understanding that difference between using a high-powered laser or laser that has infrared, or think infrared saunas, those infrared lights are being absorbed by the water molecules, and it causes the water molecules to oscillate and vibrate, and as a result, photothermal reaction. So last point here on the slide, considering that low quantum energy per photon, so again, getting into quantum physics here, but just a key few numbers that we want to take away. 785 to 905 nanometer range, that's your infrared wavelength. So when we start getting up into those 700 plus, so when you start looking at different light therapies and you start to look at the things that we'll show you guys today, if we see that some form of device is saying that we're using a 700 or an 800 or a 900 or 1,000 nanometer wavelength, that's infrared. Same as if you're reading research. If you're reading the devices they were using those sorts of numbers, and we've got a slide that breaks this down later on. That's the infrared range. Now, when we look at that infrared range on the slide, 785 to 905, that photon in that infrared wavelength, so if it's 785, that has an electron volt of 1.52, versus a 905 nanometer has a 1.37. So what we're seeing is the higher that nanometer goes, so 700 to 800 to 900 to 1,000, the higher that goes, the electron volts, so the energy within that light, gets less. So as that wavelength gets bigger, the electron volt, the energy within the light, gets less. And now why that's important is that apparently does not induce direct photochemistry. We've already alluded to the mechanical stuff. But the reason it doesn't induce photochemistry, as the minimum quantum energy is in the order of 1.7 electron volts, which is the color red. So the red wavelength, when we get into the 600s, that appears to be the minimum energy that we need to have that electron volt level of 1.7. Now, keep building on that idea of this photochemistry and quantum. In principle, one photon can activate one enzyme molecule, which in turn can process many thousands of substrate molecules. If the effect of one photon can be amplified biologically, then one does not need a lot of photons to produce an effect. So again, the idea that we're really starting to push here is understanding with light therapy, it's not about having heaps of photons. It's more about getting the right color of light that has the right amount of energy, so electron volts, to be able to create that physiological effect. So the last point, one just needs to find the proper wavelength of light to stimulate the proper enzyme, which in turn will stimulate the beneficial therapeutic effect. So the color of light through the literature is basically what's coming to the forefront here of choosing the right color of light to have enough electron volts, enough energy to be able to have that appropriate effect. But it has to be absorbed too. And that absorption process is obviously an important part of this. So the physical mechanisms of laser, I said to you that chart that we had there before about infrared, and we can see our visible light spectrum there on the right. So we can see violet on the left of the picture, showing you a nanometer wavelength of about 400. Whereas you go down to infrared, down on the far right, and we can see that's getting into that 800 range. So when we start to get into that, mid sevens and up, we're starting to see our infrared. And down the bottom, we can see the energy within each photon of that light. So for violet colored light, so a wavelength of 400-ish nanometers, we can see it has a photon energy of about 3.1 electron volts. Whereas you look down at the other end at the infrared, we can see about 800 there, 1.5. But in order for visible light, or let's just say light, because obviously infrared you can't see, but for light to be able to have a physical effect, we need a photochemical effect. Important to clarify that. For it to have a photochemical effect, we need a wavelength that has enough photon energy or enough electron volts of basically 1.7. So we can see here, looking at the picture on the right, 1.7 is going to be obviously in between the red and the infrared, somewhere in that 700-ish range. All right, so high 600s, 700 range. But we can see that as we go to the left and as we get towards violet, the more energetic the light, violet, has more energy, more electron volts. So the information there on the left of the screen, the energy of an individual photon depends only on the wavelength. So how much energy has got to do with color, not what a company says, my laser, my LED, my whatever, has this much power. That power has nothing to do with the mechanisms of light. Okay, it's about the energy, it's about the color. Right, not about how much power is listed in the device. All got to do with the color of the light. So therefore, photochemistry is dependent on the photon energy, the electron volts, not power. So it's not about wattage. All right, so you hear watts on the device. Watts is the same as power. All right, so we've got to, again, the terminology is where we get a lot of confusion when it comes to the various forms of light therapy. So when you think of power, you think of watts. When we're thinking about energy, we're thinking about the color of light, the electron volts. So as we can see from the picture on the right, violet photons have more energy than green photons, that have more energy than red photons, that have more energy than near infrared. So the key concept, a minimum photon energy of 1.7 electron volts is required to cause electrons to jump to higher orbits. If you guys go back to your chemistry or your physics and you remember the whole periodic table, and you had obviously the atom with the circles and the rings around, and we had the 2, and then we had the 4, and then we had the whatever. But in order to make those electrons jump the orbits, in order for that to take place, you need 1.7 electron volts for that energy transfer to be able to take place. So you cannot make up for a lower electron volt by increasing the wattage or power to trigger the same reactions. It's got to do, again, with the color of the light, not the power of the device. So as we've said before, low-level laser effects involves the mitochondria. The electron transport chain creates an electrochemical gradient that leads to the creation of ATP, as we talked about, which we know as oxidative phosphorylation. It occurs in the mitochondria in both cellular respiration and in photosynthesis. Obviously, think of the same process occurring with plants and plants absorbing the sunlight. So this process cannot happen with infrared lasers. As we said, infrared's absorbed by water, and it makes water molecules oscillate, creating a photothermal effect. So the electron flow starts at complexes 1 to 2 to 3 to 4, and then makes ATP once it reaches complex 5, known as ATP synthase. Low-level laser therapy targets complexes 1, 2, 3, and 4. So if you look at the picture there, what we can see is complexes 1 and 2 are affected by violet light. Complex 3 is green. And where we look at the majority of the research since the 1960s has all been done on red visible light, and that affects complex 4. So what we're going to start to see in a bit is the function of the mitochondria and the importance of different color lights to influence different complexes. Because if I've only got a red colored visible light laser, as you can see, I'm only going to influence complex 4. Now, what if the problem in the mitochondria is happening at complex 1, 2, or 3? The red's not going to influence that. So sure, the red's going to help, but this is where you might hear the stories of people saying, oh, I went and got, laser therapy or some form of light therapy, and it did nothing for me. Well, maybe from a laser therapy perspective or a light therapy perspective, again, were they using a photochemical response with low-level laser therapy? Were they using a high-powered laser? Were they using infrared? Were they using LEDs? Each of these are different. But if we just say that everyone was using a red visible light in that sort of 630 nanometer range, we know that that red light is being absorbed in complex 4. And if that patient turns around and says, the laser therapy did nothing for me, laser therapy was useless, again, the question we need to ask is, but what if the problem was at complex 1, 2, or 3? And then that can somewhat start to make sense as to why that light therapy may not have worked for that person, because the problem was happening earlier on in the mitochondria. Again, just some food for thought there. So again, the key concept here is complex 1 is considered to be a rate-limiting reaction. So if there is dysfunction at complex 1, it will affect all downstream complexes. So because that is the start of that whole electron transport chain, if there's an issue at number 1, then those issues of dysfunction are not going to be able to progress. Well, the function of the electron transport chain is not going to be efficient as it moves through because it's already been dysfunctional at the start. So we can only impact complex 1 and 2 of the mitochondria with a violet wavelength, as it's the only one with sufficient electron volts or photon energy to trigger the necessary electron jumps. Now, each complex, again, just a different picture of that mitochondria there. So complex 1 to 4 of the electron transport chain, again, as we know, is a crucial role in producing ATP. If one or more of the complexes are inhibited, ATP production will be limited. So the one thing I always think about when, from a condition point of view, and we think of ATP is obviously all of our cellular energy, but the idea of like the chronic fatigue syndrome is just like one that immediately stands out in my mind of ATP production, fatigue. Now, there's not many research articles out there that are specifically looking at photobiomodulation in chronic fatigue, but it's the one condition that sort of makes sense. If we could have something that can help our cells produce ATP more efficiently, then we could have a potential physiological effect of helping that person with their fatigue, just purely from a physiological mechanism here with, again, very limited evidence that's out there on photobiomodulation in chronic fatigue. So we've said complex 1 and 2, violet. Complex 4 is red. And as we alluded to on a previous slide, complex 3 is green. So there are specific wavelengths, so specific color of lights that have specific reactions within the mitochondria. So which wavelength or device is best? Well, it depends on the condition, which we'll start to have a little bit of a look at in a second. So each wavelength affects the electron transport chain protein complexes differently. So we said violet for 1 and 2, green for 3, and red for 4. And so we've got a wavelength-specific unique photochemical effect. Each wavelength or color of light has a specific photon energy, so it has a specific electron volts, and certain electron volt energies are required to trigger those specific reactions. So as we said, the violet is more energetic. So as we move towards that visible light spectrum, the blue has the most energy. And then as we move down, we get to the green and so forth, get down to the red where we get less energy. So let's break these specific sort of reactions down a little bit. So this is a nice little slide that came out of Nature Reviews and just talked about mitochondrial diseases and what types of presentations or what type of conditions someone could come to you with because of a mitochondrial problem. And so if you look at a lot of these here, like if we go just on the right side there, so peripheral neuropathy at the bottom right, but we can see exercise intolerance. That makes a lot of sense because if I'm not producing ATP effectively, then my muscle cells are not able to obviously produce energy to be able to keep contracting and relaxing that I need to be able to do from an exercise point of view. I'll start to produce lactic acid as a byproduct as I'm trying to go into that lactate anaerobic system to try and help keep producing ATP. And then I start developing that exercise intolerance. It makes sense. But have a look over on the bottom left. And the one that's fascinating there for me is not bone marrow failure, short stature, but kyphoscoliosis. So the idea of like this adolescent idiopathic scoliosis where we don't know in a lot of cases what's causing it, but here we are, that could be caused because of a mitochondrial issue. And we've got a whole other list of different mitochondrial dysfunctions there that are listed in that particular Nature Reviews article. And again, all these references are there for you guys, and all the references are in the final slides at the end of the presentation. So you can go and have a look at these for further input. But where we get really interesting, so the impairment of the electron transport chain and mitochondrial function. So look at the title of the article, Drug-Induced Mitochondrial Toxicity in the Geriatric Population. A common example of electron transport chain inhibition and impairment of mitochondrial function comes from over-the-counter medications. So, of the most commonly used prescription and over-the-counter drugs in the US for older adults, many are known to cause mitochondrial toxicity, such as our cholesterol-lowering drugs, our pain medications, heartburn medications. So just look at the pain medications there. Obviously, your Lipitor, Crestors, we see these a lot for the cholesterol stuff, but this is obviously looking at older population, but let's just look at all populations, aspirin, Tylenol, or Panadol here in Australia. And let's just have a look at that further. So if we look at this study, first study to describe the inhibition of mitochondrial complex 1 as a common feature from NSAIDs. So taking Nurofen, ibuprofen, aspirin, these NSAIDs, think about how many patients that you see that are on these medications. And this particular study has shown that these NSAIDs that we all just commonly use, go and pick them up in Woolies and Coles, over the counter, obviously at the pharmacy. So inhibition of mitochondrial complex 1. So these drugs are starting to show they're having an effect on your ability to produce energy. So the potential long-term consequences of these drugs. So complex 1 is considered to be a rate-limiting reaction, as we alluded to before. If there's a dysfunction here, it'll affect all downstream complexes. You can only impact complex 1 and complex 2 with a violet wavelength, as it is the only one with sufficient electron volts, again, to trigger the necessary electron jumps. And you're going to see me put a few of these little drop-down things in the slides over and over again, just to keep emphasizing these points of how the various electron transport chain complexes and light influences those. So takeaway here, and I don't want to get into too much of the whole litigation stuff, but in the US, there's a big thing with Tylenol right now, and people putting in like the big class action and suing this because of Tylenol's effects when pregnant mums were using it and it influencing the mitochondrial aspects of their unborn child. So this idea of understanding how these things work. So mitochondrial complex 1 from the use of NSAIDs. If we look at the brain now, And again, study done obviously in animals, so not a human study here per se, but brain is highly dependent on ATP, and most cell energy is obtained through oxidative phosphorylation. As we know, needs to go through that whole electron transport chain within the mitochondria. The results showed that paracetamol administration inhibited the activities of complex 1 and complex 4 in the cerebral cortex. So again, from a light therapy point of view, if you wanted to go after this, if you don't have violet and red, you will not impact both of those areas and you'll get a lesser result. So if we're seeing these drug-induced effects of mitochondrial toxicity, and we've already seen that NSAIDs, aspirin, ibuprofen, paracetamol here, we're seeing that complex 1, and obviously in paracetamol, complex 1 and complex 4 are being affected. If we are going to be helping a patient who's potentially got these mitochondrial toxicities from drug use, then we need to be using both colors of those lights. So as we get 1 and 2 are impacted by violet, complex 4 is impacted by red. If you don't have those multiple wavelengths in your arsenal, you're missing out on impacting the whole electron transport chain. So here we go with one of the important parts of mitochondrial dysfunction, and it is a vicious cycle, the vicious loop of it. Unlike other organelles, mitochondria contain their own DNA and must be either tolerated or corrected by, obviously, the human body physiology, obviously animals, same thing, to preserve mitochondrial function. Importantly, replication of damaged mitochondrial DNA can lead to stalling and introduction of mutations or genetic loss. And then where it gets interesting from this article, dysfunctional mitochondria survive longer than healthy mitochondria. In this way, some types of dysfunctional mitochondria build up and eventually become more common than healthy ones. So this area here, again, understanding different colors of wavelengths, and I'm going to make a pretty bold statement coming up in a slide soon, but understanding our wavelength colors to influence the different complexes. So mitochondrial disorders comprise a molecular and clinically diverse group of diseases that are associated with mitochondrial dysfunction, leading to multi-organ disease. However, the therapeutic approaches for these disorders have lagged behind our understanding, and treatment options for them are limited mainly to symptom-specific therapies and supportive measures. Here comes bold statement. So while in the pharmaceutical industry, we're searching for a pill solution for these things, and we can't find it. So we can already see how we've got certain medications influencing complex 1, and then we can see how we've got a complex 1 and 4 being affected by a different medication. So we're going to have these different effects. But what we can see here is when we're looking at mitochondrial health and function, we're starting to understand that light therapy is allowing us to enhance our overall mitochondrial health. So to be a laser Jedi master and impact complex 1, 2, and 4 of the electron transport chain, you will need 2 true collimated laser wavelengths. Now, I just want to emphasize the one point here about this collimated laser wavelengths. We saw it before when I just chucked that over here on me. So as we can see with those beams, if I get that further away, obviously the beam obviously gets a bit longer. And so those beams of light, they're collimated. And the reality is when we start to talk about light therapy, I'll just go as far to say that's what you're paying for when it comes to a lot of the lasers, the ability to collimate that light, because then that's directing all of that photons in that one pinpoint line, as you saw with that beam. Whereas above me, I've got LEDs above me right now. They're actually having a therapeutic effect. But the problem is because it's so broad, it's not collimated. When it's coming down onto my head, it's broad, not collimated, so you have more scatter than you do absorption. So to enhance absorption, we want collimated lasers versus LEDs in terms of better absorption effects. And again, this is the cost-benefit ratio. Obviously, LEDs work. They do work. They just don't work as well as lasers because of needing them to pretty much be sitting right on the skin. And then you go the next thing of, well, you're not going to get so much absorption. We know like the hair creates scatter. I don't have to worry about that, of course, for me. But putting an LED and having hair on your head, if you're doing transcranial work, like hair creates scatter. So I'm not going to have as much absorption there. So we can see this is the little things we're going to have to weigh up with the mechanisms of the different light wavelengths that you're using, the collimation or lack of collimation, and the power, of course, and the biphasic dose response. I'm not really covering that in this presentation, but that's a huge one. I've got, I think, 2 slides maybe on it coming up. But these are our little bits of understanding when I think about using a particular device. What is it that I want to use and why? And then, of course, understanding the research and the evidence for the different mechanisms behind photobiomodulation. So the wavelengths and the evidence-based therapeutic effects. So if we look at osteoarthritis and the role of the mitochondria in osteoarthritis, recent ex vivo studies have reported mitochondrial dysfunction in the human osteoarthritic chondrocytes, and analysis of mitochondrial electron transport chain activity in these cells show decreased activity of complexes 1, 2, and 3 compared to normal chondrocytes. So points here that I keep trying to emphasize is no matter how high you ramp up the power or the watts of the device. So when we start to look at, that you get on the back and or the marketing of it, and it's like, this is a 50-watt device. Plant the seed here right now. Watts, transfer it to joules. And so when we look at light therapy, there is a hormetic response, the biphasic dose response, the Arndt-Schultz law. I just said the same thing 3 different times in 3 different ways. When we look at light therapy, watts equals joules, and our general rule is you do not want to be going over 10 joules per centimeter squared. So if 1 watt equals 1 joule, if you have got a 40-watt laser and we don't want to put in more than 10 joules per second, can we see the issue here? So like if you've got a 10-watt laser and it's 10 joules per second considered sort of the best stimulation effect, that laser's going on for like 1 second. Now, what the research shows is that you are better off having lower doses of light being applied for longer periods of time to get those joules in, and not this idea of having it on for a second and that's all the treatment that you need. So understand that ramping up the power or watts of an infrared or even a visible red laser, you cannot impact complex 1 or 2, because remember, red is absorbed by complex 4 and infrared is absorbed by water. It's not even absorbed in that mitochondrial electron transport chain aspect. So we said that osteoarthritis was complexes 1, 2, and 3. So as a result, to influence this, you would need to have violet wavelengths to be able to impact complex 1 and 2. We get really interesting with this. I mean, in America, this is biggest burden on the healthcare system is neurodegenerative diseases. And so when you look at this particular article, mitochondrial dysfunction and biogenesis in neurodegenerative diseases, if you look at the picture on the left here, Alzheimer's dementia, we've got Parkinson's disease, Huntington's disease, and ALS, amyotrophic lateral sclerosis. And you can see here the mitochondrial relationships for each of the particular devices. Now, I'm hoping my cursor's showing up on everyone's screen, but I'm circling around here Alzheimer's, and we can see here has a relationship with complex 1, complex 3, and then complex 4. And then you can go through them. Parkinson's disease, complex 1, complex 4. Huntington's disease, complex 2, complex 3, complex 4. ALS, all 4 of them. So as we can see with them, they all go to red. All right, they all go to red, the complex 4 here. But we can see that complex 1 is involved in pretty much all of them, with the exception of Huntington's disease. So again, complex 1 impacts the tau and alpha-synuclein. Complex 2 and violet impacts the mutant Huntington gene. And then red impacts complex 4, which is your amyloid beta plaque component. again, based on this particular literature article here on neurodegenerative diseases. If we look at aging, skeletal muscle, from an aging perspective, statistically significant decline in the activities of complex 1, 2, and 4 of mitochondria isolated from the vastus lateralis muscle in 29 patients aged from 20 to 90. In the brain, age-related effects on the electron transport in the brain tissue seem restricted primarily to complex 1. Obviously, looking at different rhesus monkeys showed a statistically significant decline in complex 1 activity relative to that in complex 4, but to a lesser degree. So what we can see here, another one talking about the electron transport chain, ATP production. There are theories out there about humans have only got potential to live to whatever is 120 or 130, whatever it may be. And it's just basically the idea is that because your mitochondria are not going to be able to produce ATP anymore. So the idea of us aging, deteriorating, and this relationship with your mitochondria and the electron transport chain. Again, some research articles here on the fact of skeletal muscle and the brain and aging processes and showing that these complexes being affected. So not only are we talking about some things here, we've talked about osteoarthritis, we talked about obviously medication toxicity, talked about neurodegenerative diseases. We just had a look at aging in relationship to those mitochondrial components. But when we look at that 405 nanometer wavelength, which is the wavelength that's in this particular device, and the impacts of violet onto complexes 1 and 2, starting to understand those mechanisms. So flavin mononucleotide is a component of complex 1, just like cytochrome C oxidase is a component of complex 4. So flavin mononucleotide and flavin adenine dinucleotide, which is in complex 2, flavin literally translates to blue light absorbing, or means blue light absorbing. So the flavin component of complexes 1 and 2 is what's absorbing that violet blue light. And then cytochrome C oxidase is what's absorbing that red light. Again, pictures over here on the right are just showing you those wavelength components, the electron volt components that are involved in that electron transport chain absorption. So the 420 and 540, as you can see, I'm trying to go through whenever they're talking about a particular complex or color, and I'm trying to color code them for you. So 420 nanometers, which is that violet wavelength, 540 nanometers, which is the green wavelengths, were more effective in stimulating osteoblast differentiation compared to the red 660 and infrared 810. And then there's also been nitric oxide from COX. So one of the mechanisms, if you go back to having a look at, say, my, pretty sure it might have been webinar 1 from last year, where we break down the mechanisms of how this whole process of light therapy works. And we looked at a lot of the research behind red light into cytochrome C oxidase. And the idea here is obviously that nitric oxide binds to COX, but what seems to happen is that the red light goes in, dissociates the nitric oxide from the COX, which then allows oxygen to come back in to create that appropriate process in the electron transport chain. So when we look at this, evaluated light-mediated release of nitric oxide from COX, or also COX, as we said, is sometimes shown in literature as CCO, so cytochrome C oxidase or COX interchangeable. So using 447 nanometers, 532, 635, and 808. So exposure to 447 nanometer blue colored light led to significantly higher release of nitric oxide. The immediate increases in cellular nitric oxide levels depended greatly on the substrate sources of electrons entering the electron transport chain. So just remember the mechanism here is you dissociate the nitric oxide, allows oxygen to bind to COX to be able to create a better response. So the idea there is they're showing a significant higher release of nitric oxide means that we are going to get a better response because of the nitric oxide being released. So documented that irradiation with 400 nanometer violet laser increased maximal respiratory rates 53% in the myotubes, which are skeletal muscle fibers formed by the fusion of myoblasts during a developmental stage. So again, more pictures of information forming that the violet light, more energetic light, impacting complexes 1 and 2 earlier in the electron transport chain having better effects. And if we talk about some of even the research that Erconia has done when they compared their own devices using their red laser versus using their violet and red, the results that you got from violet and red far exceeded red on its own. And we start to understand some of these mechanisms now, starts to make a little bit more sense as to why these effects are taking place. So another study investigated the effects of 405 and 665 on reactive oxygen species. Now, remember, reactive oxygen species are considered key secondary messages within our cells. And so after blue light therapy, blue light irradiation, intracellular reactive oxygen species levels were increased in all types of cells. In contrast, intracellular reactive oxygen species generation was not observed after irradiation with a red laser or near infrared laser. Now, this one particular study, and again, this is the problem, we can collect studies, and indeed I've done the same thing, get research, present research, and you can manipulate stats, you can manipulate research to support your case, because I can show you other studies where we can see that red laser does help with intracellular reactive oxygen species, because this has been the whole basis of light therapy since it's been getting research since the 60s. So understanding those mechanisms, but what I would generally have to say is that using blue light, you're going to get a better response again because of those energy effects of that color of wavelength. Some of the stuff in here, it starts to get fascinating. Antimicrobial effects of the, now that makes sense. Think of ultraviolet light. All right, so we use ultraviolet light in hospitals, okay, for treating various skin conditions, etc. So if we're looking at blue and violet light, Erconia themselves have done a study on nail fungus and acne using the violet red, and they showed that for toenail fungus, 1 treatment per week for 4 consecutive weeks, just 12 minutes of applying the laser across the toenails, toenail fungus decreased from 23.9% at baseline to 13.9% at 36 weeks, and then 62% had achieved completely clear nails at that time. Each parameter showed further improvement after 48 weeks with no evidence of reinfection following those 4-week treatments. So Erconia did that study using violet. Again, their particular study is also out there for acne as well. But this one's an interesting one. So the research team concluded that 405 nanometer light has antiviral activity. I mean, just look at the title of that article. That's a very, that would have been a very heated topic going back a few years ago to bring up a study like this. So some wavelengths can decrease microorganisms such as blue, ultraviolet, or violet wavelengths. We talked about that using those ultraviolet lights in hospitals even. So blue light or different wavelengths has been reported to have positive effects on our immune system. It can be used for antimicrobial photodynamic therapy for bacterial, viral, parasitic diseases. Now, this is the point, and I'm going to emphasize it again with one of those red drop downs, that the wavelength used for these antimicrobial components for viruses, bacterial, etc., like the toenail fungus for acne, the wavelength used 405 nanometers, so violet, with the power of 1.5 to 2 milliwatts. And this article stated this, which should not exceed that mentioned power due to the high energy of that blue wavelength, the violet wavelength, and the duration of the laser radiation, according to these factors, should be from 10 to 20 minutes. So this study for parasitic, microbial, viral, bacterial effects, blue violet light, very low power, which is what one of these is, okay? Very low power for a longer duration. So not the idea of like 15 watts, 30 watts, and putting it on there for less time, okay? So the goal with this type of viral activities to reduce the inflammation, this is in the COVID particular study, reduce the inflammation of the lungs and increase the amount of nitric oxide that will increase the body's immunity and improve oxygen delivery to blood and tissues. So this idea of using blue light therapy to treat COVID. But the point there, the third dot point down, is what we want to emphasize. Note the warning to keep the milliwatts low due to the high photonic energy at 405. If you see a 405 nanometer device that has high power wattage levels, and this is where we get to the legalities now, please understand that there could be serious risks with increased power at this wavelength, because additionally, there's no FDA clearances for any violet laser over 5 milliwatts currently. That is the only one. That's the only one. And there are devices out there that are using violet colored light, because obviously this is where the research is going, but their devices are high powered and they have no FDA clearance for doing these things. So keep that in mind from your own scope of practice, APRA registration, etc. The devices that we may choose to use based on understanding our science and our literature. Now, a point here again about dosages. A 1 joule dose had the best effect for supporting interleukin-10 levels. Increasing the dosage caused there to be no effect on interleukin-10. So 1 joule, I said to you guys before about the idea of 10 joules per centimeter squared appears to be, excuse me, the absolute sort of sweet spot, the highest sort of dosage level you really want to go to when we look at all across the literature. And this one's just showing that 1 joule had the best effect. So if I've got a 30-watt device, if I put that on someone, I've immediately applied 30 joules in a second. All right, so I've already exceeded that joule dosage by 30 times the amount. So again, starting to understand that joule dosage with our power. Interleukin-10 is important to modulate positively for prevention of the interleukin-6 cytokine storm. This again shows that hormetic effect or the biphasic dose response or Arndt-Schultz law. An increased dosage does not mean better results and can actually cause the opposite effect of what you're trying to accomplish. And again, we're trying to find that sweet spot or Goldilocks zone. General rule is we don't go over 10 joules per centimeter squared. And in these particular cases, we've got specific research saying this is the dosage that's best for this. So that's what we obviously want to be following if we're trying to target this as a particular therapy. So the red wavelength that's in with the Oconia laser, 635 nanometers, as we've mentioned before, cytochrome C oxidase. If we're using a red colored laser, it's photochemistry. If we're using infrared, it's that photothermal mechanical effect. So Oconia lasers have done chronic heel pain studies, 2 treatments per week for 3 weeks, so 6 total treatments, just 10 minutes applied, as you can see, to those areas of the foot, and obviously decreased the score from basically a 7 out of 10 or nearly 70% at baseline to a 7 out of 100 at the 12-month follow-up with just the exposure to the red colored light. And then the chronic low back pain study, 8 20-minute treatments applied to the lower back region, 2 treatments per week, 3 to 4 weeks. Very fortunate here. I will do a bit of a shout out here. So the primary investigator is a good friend and colleague of mine, Trevor Berry, who we're going to be very fortunate enough to get back into Australia this year in May, where Trevor and I will be doing 2 hands-on weekends together. And just dropping a bit of a hint, we will be going into the exciting realm of adding the green color wavelength. Been focusing on obviously violet and red with this device, but obviously you've heard me allude to complex 3 in green. So we'll be adding some of that science and knowledge into it with our understanding of those 2 hands-on modules. So very excited to have Trevor out here for that. But Trevor was the primary investigator in this, and they did 2 treatments per week for 4 consecutive weeks. And they showed that they could get their pain scores decrease of 36.6 for the low-level laser-treated subjects versus 8.9 for the sham. Now, what was the sham? The sham was using a 635 nanometer LED. So it was still laser therapy. And I'll show you the graph. So people still got better. They just didn't get as good a results as they got from the laser. So this is where it got really interesting, the 12-month follow-up that they ended up doing. After the initial 8 sessions were finished and the initial benefits obtained, and obviously those results that we showed you on the previous page, on average, patients experienced an additional decrease of pain of 17.48% by the 12-month mark with no more treatments. Now, that was just for the laser group. So let's have a look at this in the next slide. But this is where this one's important. I'm a very research and science-based person. And when it comes to Erchonia, these are the people who have done the majority of the research as it relates to low-level laser therapy, LLT. So they invest their money in going into doing research to find out if their products work or not. And so I generally do have the throwaway line. If LEDs or high-powered laser was the way to go, they would have made one. And basically the evidence is all pointing towards low-level laser therapy is the way to go over high-powered, over LED. And again, this company has put their money where their mouth is and done a lot of research. So no other laser company has shown the results through double-blinded studies versus an LED placebo. No other laser has an FDA clearance for chronic low back pain and chronic heel foot pain. So again, before you buy any laser, be sure to verify what they are cleared for and if they have research to back up their product. And again, which is why I have attached my cart to the Erchonia horse per se. But that's the study on the low back pain. And as we can see at the top, we've got our black line, which is obviously the solid black line, I should say here, is the laser group. And then our dotted or hashed line here is the LED group. So we can see the LED group still got better. All right, still got better. Not as good as the laser. But when the treatment ended, the laser group continued to get better, whereas the LED didn't. And you will hear stories about this too when it comes to some of the other LED devices that are out there, where you'll hear people in their own YouTube webinars and seminars or Instagram clips like, yeah, when I was using this LED device, I was getting really, really good results. And once I stopped using it for a couple of months, I started to regress. But as soon as I started using it again, I got the benefits again. And so this idea that we can see here with this graph, and you'll often hear little case study testimonials report the same thing as it relates to the LEDs, that once you stop using it, you start to get a bit of a regression back towards where you were. But as you're using it, you do get those improvements. So many companies claim to be better because they have higher wattage, yet they do not have a single quality blinded study to support that claim. It actually runs counter to the bulk of the research. So hopefully we've given you a bit of an insight there into mitochondria and wavelengths, obviously a very short 1 hour time. More background, of course. We've got those previous modules 1, 2, and 3 from last year on low-level laser therapy and all the science. And then obviously we'll be holding some more of these things if this interests you. And of course, if we've got any questions, please feel free to obviously ask me now. I can see that we've got here. All right, so the question is, how deep does light travel before it stops having a therapeutic effect on the mitochondria? Would it be able to reach much deeper structures or reach central aspects of structures such as the center of a bone? It is a really good question. And the question here that we want to try and answer is, this is where it actually gets interesting. So the aspect of light that's now become, and I'm going to loop around here, sorry. So when we look at some of the stuff that I had shown in some of the previous webinars, is they've done studies now where they have shielded areas of the body, and then they've gone and applied the laser, say, on the shin bone. And then what they've been able to measure is actually changes in the brain by applying light therapy at the shin. So this question now, and this has been done specifically with this transcranial, because everyone's arguing, does light even penetrate the skull? Because if it doesn't penetrate the skull, how can we say it's having any effect? So the question that's now being posed is, it's more maybe the question of an abscopal effect. Abscopal meaning having an effect away from the target site. It actually comes out of radiation therapy and let's say from cancer therapy. So actually treating a primary tumor with radiation and then seeing secondary tumor sites away from that site that's been irradiated, seeing those other tumor sites start to shrink and decrease. So they're starting to show that we actually have free-floating mitochondria going around our body. And some of the theories now that we're applying laser being absorbed in that free-floating mitochondria, and it's being transported around the actual body. Red blood cells, obviously, other cells, obviously red blood cells fully mature don't have that, but our other cells that are floating around in our bloodstream, that can be absorbing the light and transporting it to different effects. So these abscopal effects is starting to be an interesting concept of the discussion. So in terms of how deep does light travel before it stops having a therapeutic effect, that is starting to go into the realms of abscopal effects and this depth of penetration. And another problem with that, as trying to answer this, is also then the idea of most of the studies, when they've looked at depth of penetration, have been on cadavers. And they've just been trying to measure how far the light goes into tissue. But a cadaver is a dead bit of tissue. And so we don't have any cellular activity going on. So the studies that we've got of brain transcranial, like functional imaging, QEEG imaging, are showing, like just by lasering through just over through the skull here, having global activation through the brain. So I can't give you an actual measurement, but the best answer I can give you is these abscopal effects. All right, next attendee. If the only thing that matters in laser therapy is the wavelength electron volt and not so much about the watts, what would make another brand better than the other if they have required wavelengths for therapeutic effect? So if I'm sort of reading the question right here, when we look at having the wavelengths and electron volts, so if we have every electron, so everyone says I've got the same wavelengths occurring. So this device has got 635 and 405. This device has got 635 and 405. If we look at what makes another brand better, it's rather looking at the brand aspect, now you start to go, now you look at the watts between the two companies. So you go, they're the same wavelengths, same wavelengths. What are the wattages? Because if I've got more watts in a particular device, now those watts, that's the joules and the energy that goes in there. So that's that whole biphasic dose response. So the idea of this device uses milliwatts, whereas another device may use watts, and start to go into the realm of being like a class 3 or a class 4 laser. And then as we know, those lasers, when they're on people's skin, run the risk of obviously burning the skin. So the classes of laser go up higher based on the risk to skin burn and obviously eye damage. Then the next thing is going to be the collimation. So if you were to hold a laser or light that had the same wavelengths, but if you held it up above whatever it is you're pointing it at, and you start to see it get like blurry and fuzzy, it's not truly collimated, so you're not going to get as much absorption. So the best example, the best sort of answers I can give you there is we've got to look, all right, yeah, the wavelengths are one thing. Then I've got to look at the wattage, so that way I'm understanding my joules for my biphasic dose response for therapy. And then I want to look at the actual light. And when I'm pointing that light at something, am I seeing a true collimated beam, or am I seeing it start to get broad and fuzzy and it looking a bit more like an LED coming from above me? Because then that starts to make that light work more like a high intensity heat lamp. So I hope that answers that question. Are there any contraindications to applying low level laser therapy when there's any prosthetic devices hardware present? It depends on what you're saying with hardware. So contraindications, of course, is the whole pacemaker stuff. You're not going to apply laser over the pacemaker aspect. Now, putting low-level laser therapy over a knee replacement, hip replacement, that's fine, but I wouldn't be putting a high-intensity laser, high-powered laser over the top of it. So our contraindications, pacemakers, Like tattoos. You can't put a high-powered laser over a tattoo, it burns them. So low-level laser therapy, you can go straight through a tattoo, no problem. Again, it's just understanding those components. Obviously, not flashing lasers into people's eyes and staring at laser in the eyes, not using laser over pregnancies. Now, the open wound one's starting to become an interesting one because obviously some evidence with that to maybe support wound healing. So the devices, no for pacemakers, prosthetic devices for true low-level lasers, that should be fine. How effective is low level for carpal tunnel syndrome? If we have a look, it's either in my webinar 3 or in my one-day seminar that we've done. We've got a collection of protocols that we would use for carpal tunnel syndrome. Just trying to think off the top of my head, I think there's a randomized controlled trial on carpal tunnel syndrome to show the effectiveness for it. I think that's in webinar 1, if you wanted to go and have a look at that in terms of that research article. Just sorry, I can't remember that one off the top of my head. And is there a difference in the 1 Hertz activation of mitochondria as opposed to, all right, so frequency settings, all right, the Hertz, where I want to go with Hertz, if we're going to go down that, that's all about pulsing. So we can change how the light is pulsed. So if people are still online here, what you'll see is, so the violet light is set at 1 and 10, and then the red's set at 40 and 60 here. So if you have a look on my face here, you'll be able to see the actual light pulsing on my skin. Hopefully you guys can see that okay. And so that pulsing, when we look at the Hertz and mitochondria, I want you to think of it more as the suggestions, especially when it comes to brain and the transcranial effects of light therapy. We know that the brain has certain wavelengths that it tends to work better. So as an example, the vagus nerve loves 10 Hertz. It seems to fire at 10 Hertz. There's research to show the Achilles seems to be about 3 Hertz. So when we start to look at this idea of Hertz, I want you to think about more of the actual tissues that we've got evidence. And again, I'm big on the evidence. So when we look at what, and the statement here is 1 to 10,000 Hertz here, I can't think of a protocol that I ever put in my laser that uses 10,000 Hertz, like ever. The study, the stuff that I would use, the highest I've got in there is one that goes to 60. And again, that's for brain frequency things. So I wouldn't be using like 10,000 Hertz off the top of my head for anything. So the example is, does it affect treatment time for safe dosages? The Hertz there's not going to affect treatment time for safe dosages. It's purely got to do with potentially trying to enhance the actual tissue that you're targeting by trying to match the frequency of that tissue to the Hertz. I hope that makes sense. It's all got to do with the watts, so your power, which is involved in the joules. That's the primary in terms of treatment time. All right, it's all about the joules per centimeter squared, which has the relationship to the watts of the device as to how long that can be on. That's what affects your treatment time. Where can we find your old webinars? Thanks. Anne and Sherelle will be able to help direct you to the old webinars, which are basically on YouTube. They're available on YouTube, to my knowledge. And so obviously as part of this, all of our references for what we talked about today obviously are available here at the end of the presentation.